Welcome everyone to the March webinar in the Cancer Genomics Cloud webinar series. My name is Sarah Fisher and I am a community engagement manager working on the Cancer Genomics Cloud. And I am very excited today to introduce two awesome speakers, young scientists who have recently published a very cool paper, Dr. Ayse Keskus and Isabel Rodriguez. Ayse received her PhD from Bilkent University in Turkey, and her research interests include complex structural variations in cancer genomes. And Isabel received her Bachelor of Science in Biochemistry at Florida International University, and her research interests include cervical, breast, and pediatric cancers, as well as long-read sequencing. So today they're going to be speaking to us about their recent publication, Insights into the Mechanisms and Structure of Breakage-Fusion-Bridge Cycles in Cervical Cancer using long-read sequencing. And I will cease my screen sharing and cease my talking and turn it over to Dr. Keskus. You're still muted.
Yeah, no. Hello all. Today we are going to talk about our findings regarding to the breakage-fusion-bridge in cervical cancer cell lines. And I will be particularly describing Severus, a tool that we developing and we use to identify breakage-fusion-bridge cycles in those cell lines. So to start with, structural variations are large alterations in the genome. They're heterogeneous by nature, and the size of an SV can vary from 50 base pair to the entire chromosome arm. And they can be very simple, like deletions, duplications, insertion, or inversions, or they can be complex, which is defined by multiple breakpoint pairs. In this case, this chromatopsis and chromogenesis are the two very well-known examples. In chromatopsis, the segments are reshuffled themselves in the chromosome arm, and chromogenesis is amplification of the multiple segments and in the chromosome arm. Although the number of SVs is much smaller compared to the SNVs in the genome, the functional impact is much higher, especially in the complex events. Therefore, they don't occur very frequently, and the one mechanism that's leading to this complex structural variations is genomic instability in cancer. I don't really need to describe it in very detail, but genomic instability can be as copy number instability, change in the end, like numbers of the haplotype, or it can be structural instability that we observe the change between different chromosomes. But more oftenly in the cancer cell, we observe the combination of both. And since the cancer cells has a lot of structural variations, they can be oncogenic drivers and they can alter therapy response. Therefore, they are widely used for patient stratification. Here's the one paper that they analyze structural variations in cancer. This is from Peacock Group. They have analyzed thousands of patients from different cancer types. This is a complex figure, but the first thing is the number of structural variations that we observe differs between different cancer types. In sarcomas and breast cancer, we observe quite a lot of structural variations, but in some others, we only observe a couple of structural variations. And each of these colored violin plots represent a different SV type. They can be simple ones like deletions, reciprocal translocations, or they can be like in this gray one, which are complex and unclassified. And if you check the number of unclassified structural variations in each cancer type, the substantial amount of structural variations are still unclassified. And if you consider the functional impact of structural variations in the cancer, we definitely need more information on the structural variations. And long-read sequencing can help can fill the gap and help to resolve some of these complex events. No, why? Because long-read sequencing has their own advantages. The first thing is simply we have long reads. In this example, we have junction from chromosome A to B, B to C, and then C to A again. With just this information, it's impossible to decide if it is a translocation, if these are happening independently, or this is actually part of a single event. But if you have a read long enough to span all this junction, let's say this is a single read, we can clearly say that the read starts from chromosome A, jumps to chromosome B, then jumps to chromosome C, then comes back to chromosome A again. Then with this information, we can clearly say that it's a template insertion cycle, and this is not a translocation or chromoplex event. And the second one is improved phasing. With long-read sequencing, we can actually phase the reads and assign reads to paternal or maternal haplotype. In this one example, we have these two deletions in a very important gene. The first one is exon 1 and second one is exon 2. And if we don't know if these two deletions are in the same haplotype or not, we cannot decide if we're going to have the functional protein from this gene. But if we can phase them and see this is one like paternal and this is maternal, and we have deletion in both maternal and paternal haplotype, so we're not going to have a functional protein. This is actually example from a real case. We observed this kind of events much oftenly in cancer datasets. And the other well-known thing is with like long-read sequencing with improved mapability. But long-read sequencing is a relatively new technology, and the tools that are developed for structural variation analysis with long-read sequencing are mostly focused on the normal genome. And the normal structural variation types that you observe in normal genome and the cancer genome is quite different. Therefore, we develop Severus, which is specifically designed for cancer genome. We can use the germline mode with a single sample, tumor only, or it can be a somatic SV caller with tumor normal pair, and it can also impose multiple sample. It can be multi-site tumor or like time series or pre-post treatment. So here's our pipeline. It inputs BAM files. It can be phased or unphased, but we highly recommend it to be phased. And the first step is we do pre-processing summaries that I'll talk about in a bit. And then we identify structural variations in those BAM files. The first step in pre-processing of the reads are how to handle VNTRs. VNTRs are repeat containing regions that are low complexity regions. Therefore, the alignment inside the VNTRs, especially with read with indels, is highly unstable. Especially in cancer, we observe a lot of mutations in those regions. The standard approach for handling VNTR is just will fail because of the imprecise length and position of structural variations, just you can see in positioning of this deletion or this insertions in here. But how we are handling in Severus is we are combining indels for each read inside the VNTR region and uniformly represent them inside the VNTR region. That allows us to resolve this VNTR, and we can also identify mosaic events. To check if that worked, we generated the synthetic CHM1, CHM13 dataset. CHM1 and CHM13 are widely used, well-studied, normal haploid cell lines. We merge CHM1 and CHM13 and treat them as tumor, and we use CHM1 only case as normal. A structural variation occurring only in CHM13 will be our truth set. And we developed MINDA to compare SV calling from different tools. And here are our initial results. The first three is the Illumina callers. and the last four are the from PacBio sequencing. So we also use SNFLs2, which is the state-of-art method currently for the long-read SV calling, which is not designed for cancer genome, but we want to include this one as well. And as you can see, Severus in here that outperforms all the existing tools. But as I mentioned in the beginning, CHM1 and CHM13 are normal cell lines. So majority of the events that we observe in those cell lines are indels. That's one of the reasons why this Illumina callers poorly performed in the cell line, because they are specifically designed for the cancer genome. Therefore, we generate our own data sets. We sequence 4 different breast cancer and 2 different lung cancer cell line and their corresponding normals. We grow the cells, extract the DNA, and we sequence the same DNA extract with Illumina, PacBio, and Nanopore sequencing. We get the data, we phase them, then we run the somatic SV calling, we get the VCFs. Since we don't have the truth set, but just like we have in CHM1 and CHM13, we generated our own ensemble call list. For that one, SV is supported by at least 2 technologies, Illumina and HiFi maybe, and then at least 4 tool is considered as true positive, and the rest is false positive. We first check the consistency between the technologies. Majority of the events that we observed are shared across all 3 technology. That's further supporting the ensemble method is actually working, except H2009, which is mainly consists of insertions, which the green ones in here, and the insertions are particularly hard regions, hard SV types to detect in Illumina sequencing. And the second part, we evaluated all the SV callers, and Severus outperforms in both HiFi and anti-sequencing. For the most of the cell lines, Illumina sequencing, like especially MANTA, is outperformed SNFLs2, which is not designed for cancer genome. The one clear exception is ACC 1954, in that all of the Illumina callers performed clearly like similar way with the long-read sequencing technologies. Therefore, we further categorize, like further break down the structural variations into different categories to understand which ones that we can detect better and which one that we can't. This is a complex figure, but I'll just break it down. So in this figure, the color represents a different technology. Blue is Illumina, pink is Nanopore, and yellow is PacBio. And with insertions, it's a known thing that Illumina colors are not performing well, just because of the technology itself. But with the break-and homology, segmented duplication, and tandem repeat regions, we also observed increased accuracy with the long-read sequencing technologies. Among all of them, the Severus outperforms the existing tools for HiFi long-read sequencing technologies. And the second one that I want to focus on is this duplicated junctions and junction chain part. And these are the recurrent patterns in cancer genome, in which that we have a small segment in between the 2 different junctions, like in this one or in this one. And in this case, especially SNFLs2, which is not designed for a cancer genome, performs very poorly. So that's why we definitely need a cancer-specific structural variation calls. And again, Severus outperforms in these clusters as well. If I convince you that Severus can detect individual junctions, I can move on to the second part, which is the complex SV clustering. And for the complex SV clustering, we adopted a well-known method from ancestral genome reconstruction called breakpoint graphs. I'll skip the details for the method due to time constraints, but I want to show you a couple of examples that why we need to cluster that structural variations in the first place. So let's say this is our reference, and each of the colored arrow represents a genomic segment. And we have this deletion-like junction in here, which connects blue segment to green segment, implying this yellow segment is deleted. And if you just check the VCF, that's what you're gonna see is this yellow segment is deleted. And if you just check the breakpoints. But what if we have this junction to in a very nearby region? And in this case, this will be the outcome. And here, it is just a rearrangement between green and yellow segment, and there is no deletion. And actually, there's new junctions occurred in here. This is relatively easy example, and the inversions especially can be very complex. When we say a simple scenario for inversions, we think about reciprocal inversions, like in this case. The yellow segment is inverted and it didn't change in its own position. But it can be also an inversion with a deletion, like in this case. We again have the very similar junction, 1, 2. We also have 1, 2 in here. But with this junctions, we can say this green segment is deleted. But there is no clear evidence from just through the junctions that the green segment is deleted. It can be even more complex, in this case with multiple inversions occurring in a very close distance. And in here, the entire chromosomal region is rearranged itself. So the overall idea for the complex SV clustering is when we focus on single junction, we might misinterpret the results and also the rearranged genomic structure. We looked at clustering results from our own cell line data. So here are the results. Each darker color represents the number of SVs in complex structural variations, and the lighter ones are the simple SVs. As we expected, majority of them are actually a simple SV. Sorry. Except ACC 1954, which is a BRCA1 mutated breast cancer cell line, which has two known chromothripsis examples. And most of the complex events are actually has less than 6 junctions, but we have very complex events with 1937 and 1954. And this is one of the cases that I mentioned, a chromothripsis example in 1954. This is a known case with validated from like a fish, and we can clearly identify these examples as well. But these kind of cases, we only have 1 or 2. Majority of the events are like this one. So they're relatively simpler, which has like a 5 to 6 junction. And in this case, this is the graph of the Severus output. So in each green segments represents a genomic segment in here, and each of these colored lines are different junctions. If you define segments, like the colored segments in this one, this will be the rearranged genome. If you just follow through these lines, and it will come here and jumps here and then jumps back and then continues in here. And we're gonna end up with 2 deleted segments in this graph. And the next part, we check if it is caused any copy numbers change. We run Vahan, which is a haplotype specific copy number caller for long-read sequencing. And with this second haplotype, we see an increase in the haplotype. We see increase and decrease in the copy number in this region. So with Severus, we can identify the complex events and can resolve the complex events with the breakpoint graphs. And the last part, I want to focus on a very specific complex event case, which is a breakage-fusion-bridge. And breakage-fusion-bridge, it starts with a double-strand break, as always. And when the double-strand break happens, and the next cell cycle, when the DNAs get amplified, since cell doesn't want this double strand break. It just connects it to the sister chromatid through non-homologous end joining, and this will be the resulting chromosome. And this one has two centromere. Each of them will be pulled apart to the daughter cells. Therefore, it will cause an additional break. And the next cycle, this will happen, continue in multiple cycles. And this is the very well-known copy number profile for the structural, for the BFP, this tail line structure. That's what's mostly used to identify those cases. With Severus, we also can identify breakage-fusion-bridge cycles. And this is a one tiny example. Ayse will give more details on breakage-fusion bridges, so I'll just give the brief talk about this. So this is the one stair-like structure from telomere side. So this come here and then connects back to fold-back inversion. And then we have an additional fold-back inversion in here, and the centromeric side, the fold-back inversion occurs in here. So with Severus, we can identify fold-back inversions and breakage-fusion-bridge cycles. So as a summary, Severus can work with a single sample with the germline calls tumor normal pair and multi-sample tumor case. It outperforms the existing SV callers, and we can use breakpoint graphs to cluster and regenerate the complex SVs. And Severus is freely available in here. You can always check. And finally, I would like to thank all of our collaborators and our group Conograph Lab. And these are all the tools that I talked today. So thank you. I'll stop here, and Isabel will continue with the our findings with cervical cell lines.
Okay, can everyone see my slides? Yes, okay. Well, thank you, Ayse, for explaining all of the bioinformatic part, because I am not very good at that, and she is kind of the one that takes care of all of that. So I'm going to be focusing more on the cervical cancer part of it and what we saw in our paper, using what Ayse just very kindly and calmly explained to everybody. So to start off with cervical cancer. Cervical cancer is caused by the human papillomavirus and is responsible for almost all of our cervical cancer cases that we see. It's the fourth most common cancer worldwide, and its incidence is high in low and middle income countries. So incidence of cervical cancer depends on various things, such as population age, distribution of cancer risk factors, and socioeconomic status. Low and middle income countries have had reduced access to medical care screenings, clinics, and fundings are classified as low in the Human Developmental Index, and which have been seen to have higher cervical cancer incidence and mortality and account for 90% of the cervical cancer deaths annually. So HPV has been recognized as a preventable disease since there is a vaccine. And as seen on the vaccination map on the left, not all countries have been vaccinated at the same rate. Countries like Africa and Asia, where HPV is high, have low vaccination rates. So the World Health Organization has initiated a plan to help eliminate cervical cancer by 2030. And their goal is to decrease the incidence rate in countries by achieving screenings of at least 70% of all women above the age of 35, vaccinate 90% of girls over the age of 15, and treatment of 90% of pre-cancer patients. So to talk a little bit more about the HPV virus, the HPV virus is a commonly sexually transmitted infection, and although there are over 200 types of HPV, 15 are recognized as high risk. High risk meaning they're more aggressive and likely to develop into cancer itself. So according to the CDC in 2018, there were about 43 million HPV infections. Mainly among them were kids in their teens or early 20s. And in most cases, HPV does go away and the virus is cleared on its own within the first 2 years. But this isn't always seen. Usually the cases that do progress to cancer are seen in high-risk HPVs. So this means that HPV is not only the main cause of cervical cancer, but can also cause cancers such as vagina, penis, and head and neck cancers. And although there's no way to know exactly if HPV will develop into cancers or the mechanisms on how this happens, many advances in HPV research has helped us formulate a hypothesis in where cell lines with various HPV types have been further explored to understand the mechanism. In our lab ourselves, we have used nanopore technology to do long-read sequencing to help provide a bit more insight when we're talking about the DNA and RNA of HPV. The virus itself is a 8,000 base pair circular DNA and encodes for E6 and E7 oncoproteins, which bind and inactivate the tumor suppressor P53 and RB1. So HPV and HPV-driven tumors, the virus is often integrated, but it's not primarily part of the HPV's life cycles. So as it seems as of right now, it randomly occurs if it does, and it can lead to the formation of tumors and cancers. So with the use of nanopore sequencing, we successfully were able to map out HPV reads in both cell lines and tumors to get a more in-depth analysis of what was going on with the integration. So the goal of the study was to use 22 cell lines of sequencing both DNA and RNA. 10 of them were Korean cell lines that were not in COSMIC or CCLE cell line databases. And we got a comprehensive view of these cell lines. We looked at HLA typing, HPV integration, and HPV negative cell line, cellular driver mutations, and BFB events. We did this by just growing up the cells, extracting DNA and RNA, and then using various different techniques to do Oxford nanopore technology. So we used RNA, DNA, and different kits that they have. So our panel was about 42% East Asian, 50% were HPV-16, and over 75% were squamous cell carcinoma. We made a map of where HPV would integrate. So these are all of our cell lines and where they integrated, with the exception of our HPV negative cell line, which is not integrate because no HPV is present. So although there's no certain pattern, we did notice that some of them do integrate in what are called hotspot regions. And these regions are where genes of importance could be. So we looked at each cell line individually to see where exactly they integrated and what was going on with them. So one thing that we noticed was that sometimes during the integration, the genes near can be turned on or off. So this is called insertional inactivation, and it was seen with SNU 1005 with the RAD51B gene. So the DNA repair gene became inactivated. In SEC 154, the FOXC1 gene actually became highly expressed, and in SNU 778, the RAB8T gene, which is part of the RAS oncogene family, is also seen to be overexpressed. So these are patterns that we started seeing with the integration sites. Overall, we looked at the mRNA and we made a Z-score table. So if you look at the Z-score table, the black boxes are the cell lines and the cell lines near the integration site. So on the left are the genes, at the top are the cell lines, and next to the gene, you can actually see what cell line is integrated near. So if you look at the darker green boxes, which are higher mRNA expression, versus the light green boxes, which are low mRNA expression, you see that near the integration sites, those genes tend to be of higher expression. Further looking into expression, we looked at HT3, which is an HPV 30. It has HPV 30, which is an unknown risk. It's not seen as high risk, yet the cell line did progress to cancer itself. So we noticed that it is not only integrated near RB1, but RB1 also appears to have a mutation in HT3. So this means that it can play a progression into it becoming cervical cancer. We looked at all of our cell lines to see mutations in RB1 and TP53 since we noticed this. C33A was in HPV negative cell line, which means no HPV is present, yet it functions like HPV because it has mutation on both RB1 and TP53, which would be like if there was HPV present, because like I said previously, the virus for HPV uses E6 and E7 to inhibit these two tumor suppressor genes. Then we continued to look at our cell lines, and we noticed what Ayse briefly ended with, which was our breakage fusion bridging events. So again, to give a quick overview, breakage-fusion-bridge happens during mitosis when the chromosomes are duplicating. So there appears to be a break in the chromosome near the telomere. It has these sticky ends. They fuse together and form a bridge. When that part of mitosis pulls apart the centromeres, it's pulled randomly and it breaks. And then you have a sister chromosome that can be either longer or shorter than it's supposed to be. And this term was coined by Barbara McClintock in her study in corn. So she noticed the same thing. And yeah, so the telomere breaks, then it fuses. Then after that, there's the break and the bridge, and it goes over and over again. So that's what the normal cell would look like. And then on the right is what the tumor cell would look like. And that's exactly what the copy number plot would look like. So in our cell lines, we saw 3 different types of BFB. So we had the normal breakage, but right before the fusion step, we saw some digestion that took place, which means part of the reads were missing. So instead of just having the same read and the other read going inversely, we saw that part of it was missing, and we saw that it happened differently, and we classified these as 3 different events. So type 1 BFB would be with a breakage, a digestion, a fusion, and then there would be just the digestion and the fusion. So this would be type 1, and it would have a mismatched end. So as you can see here, you have A going from left to right, which is the normal read, the way it's supposed to go, and then B and A. So normal BFB would look like A, B, and then B, A, but since there was some digestion, we only have one of the B segments. For type 2, we have an insertion from another chromosome. So that's inserted in the middle, so it's shown in yellow. And type 3 is a rearrangement within the chromosome itself. So the purple is from the same chromosome, an area from the chromosome came and was attached right between the A and B. So looking at type 1, we saw this happen in HT3. So normal coverage for HT3 would be about 40x. And in chromosome 17, we noticed a drop in coverage that looked like a staircase. So it went from 95 to 54 to 16. And as you guys see in the reads that are rainbow, those are mismatched reads. And they clearly cut off right before it jumps from 16 to 54 and right before it jumps from 54 to 95. So this is what it would look like with the digestion before the fusion. Type 2 is when we have an insertion from another chromosome. And we observed this in SNU 1000 chromosome 2, where we saw there was an 168 base pair insertion from chromosome 7. So again, the model would look like AB, but instead this B is replaced by an 168 base pair from chromosome 7. Type 3 would be the rearrangement itself, and we notice this with SNU 682, where the WT1 tumor suppressor gene actually became inactivated because 5 exons were deleted. So instead of seeing a clear step like as we've seen in the previous 2, this one we just see a jump from 6 to 75 coverage because the exon is gone. Further looking at BFB, we noticed that we had a couple of cell lines that had an event near the same place in chromosome 11. And looking further, we saw that it dealt with the YAP1, BRCA2, and BRCA3 gene, which play high importance in cervical cancer. So these are 3 of the cell lines that we saw that had this. And we looked further to see if we had seen this in any other cell line or anywhere else. And looking at the TCGA cervical tumors, we saw that there was YAP1 amplification. And when we did the copy number, we see high copy number for YAP1, BRCA2, and BRCA3 shown in blue. And right after, there's a drop with low copy number, showing that the TCGA tumors most likely all have these BFB events. Looking at YAP1 amplification, we saw that women with YAP1 are actually diagnosed 10 years earlier on average, and women of African American descent are actually 3 times higher of having a YAP1 amplification. This data was supported using TCGA, MSK-CC, and AACR Gini. So in conclusion, we were able to use a large panel of 22 cervical cell lines and long-read sequencing. We fully characterized the sequence of HPV integrations and consensus on gene expression. we discovered that cervical cell lines that are HPV negative or integrated by a non-high-risk type had a higher level of driver mutations, including TP53 and RB1, to compensate for low or absent activity of HPV E6 and E7, and sequences at the inversion junctions of break-BFB events provide new insight into the formation of these critical chromosome rearrangements in cancer. For future directions, we are looking to examine the epigenetic consequences of HPV integration in BFB events and looking toward exploring new cell lines in PDX tumors, adding to our panel in order to reach an overall greater diversity, and explore the major molecular subtypes of cervical cancer are represented in this panel, and we hope to enhance efforts to develop targeted therapies as well as immune therapies. So I would like to thank all of our collaborators and everyone in my lab and Sierra for this opportunity. So thank you so much.